We've developed a model in which we develop a personalized vaccine for patients with myeloma that consists of their primary myeloma cells that are fused or put together with autologous dendritic cells. And the idea is that this hybridoma expresses the broad array of antigens from the patient's tumor, including neoantigens that are unique to that particular patient, and also captures clonal diversity within the tumor and puts them in the context of the immune-stimulating machinery of the dendritic cell. And so we reviewed some of the data from our prior trial in which patients who underwent autologous transplant then underwent vaccination, and we were able to show that there was a robust expansion of myeloma-specific T cells in the peripheral blood of those patients, and that correlated with a near doubling of their CR rates in the period from day 100 to 1 year out after transplant without other maintenance therapy. So we thought there was a correlation between the immune effects of the vaccine and a clinical outcome. To follow up on that, we are now leading a national study being conducted through the CTN, which is one of the oncology cooperative groups in the US, where patients are being randomized to, after transplant, undergo vaccination with lenalidomide maintenance as compared to a maintenance alone arm. And the trial has been rapidly accruing and now has nearly 100 patients enrolled. So we're very excited about seeing that data. We also reviewed some of the information about our vaccine in the context of other immune adjuvants or modifying agents that we think would have an important effect on the microenvironment. We've identified several microRNAs, including miR200C and 34A, that we think are important in modulating immune activity, and are now studying how inserting those microRNAs may affect vaccine response. And we're also looking at the combination of effector cells with our vaccine as a way of also trying to modulate immune response. We actually have looked at this in a couple of different settings. I think there's a general feeling about vaccines that they're going to be most effective when the immune system is somewhat intact, so either in patients with minimal disease or early on in the disease process. I presented some data that we recently published in acute leukemia, where patients who had achieved remission after standard chemotherapy then underwent vaccination, and we saw a profound effect on relapse rate, where 70% of those patients are relapse-free at 5 years out, which is very unusual for that particular cohort. So we think some kind of combination strategy, whether it's with transplant or other cytoreductive agents to set the table, so to speak, for a vaccine to follow, is one strategy. If we're thinking about this in the relapse setting, we're very interested in combining this with things like effector cells, like engineered T cells or CARs, where you're infusing T cells that have functional capacity and then potentially broadening their repertoire and their durability with the vaccine.